Welcome to the deep dive, where we get into the important details from complex research so you can stay informed. Today, we're looking at a really significant paper. It gives us this broad ecosystem level view of how rivers actually work. Now, the title, it's what you'd expect from a science journal. Spatio-temporal patterns of multi-trophic biodiversity and food web characteristics uncovered across a river catchment using environmental DNA. Bit of a mouthful, right? But we're going deep into the work by Rosetta C. Blackman, Florian Altermatt, and their colleagues. Our goal here is to really unpack what they learned from studying this huge river system in Switzerland.
Yeah, and the problem they tackled is a fundamental one in ecology. For ages, studying rivers meant you had to make choices. Are you netting fish, counting invertebrates, analyzing water samples? You typically could only do that at a few spots, maybe once or twice a year. But ecosystems aren't that simple. You need both biodiversity, just the sheer number of species, and the food web, how they all interact. Studying both together across big areas and through different seasons, that's been almost impossible.
Right. Impossible, logistically.
Yeah.
And you really need that joint picture for conservation, don't you?
Absolutely.
I mean, you could track fish numbers, and maybe they look okay. But if the insects they rely on, or the bacteria breaking down waste, if that whole foundation is crumbling, you're basically flying blind. You've got a false sense of security. And the big reveal from this paper, the headline finding, is that these two aspects, the biodiversity patterns and the food web dynamics, they don't track each other perfectly. They're not simple mirrors. You really can't just count species and assume you understand the trophic structure, the who eats who part of the ecosystem.
Which, of course, brings up the obvious question. How on earth did they manage it? 73 sites, a 700 square kilometer area, 3 different seasons.
Well,
That sounds like an army of researchers.
Exactly. And that's where the technology really shines. They used environmental DNA, eDNA. So for you listening, eDNA is basically the tiny traces of genetic material organisms leave behind, think skin cells, waste, that kind of thing, just floating in the water. So instead of catching every single creature, you take a water sample, sequence the DNA floating in it, and that tells you who's been living there or passing through.
And the beauty of it is how scalable it is.
Yeah.
And it's non-invasive. You're not disturbing things much. Using a technique called metabarcoding, they didn't just look for one thing. They used specific genetic markers to screen for 3 key groups all at once: fish, invertebrates, and bacteria.
Wow.
And they found a lot. 374 genera in total across those groups,
Yeah.
12 fish, 80 invertebrate, 282 bacteria genera.
So one sample of water gives you insight into 3 different levels of the ecosystem. That's incredibly efficient.
It really is.
Yeah.
Now, analyzing that huge data set required more than just lists of species. They measured alpha diversity, that's your local species richness. Basically, how many different types live at one specific spot, like Site A. And they also looked at beta diversity. That measures community turnover. So if you move, say, 5 kilometers downstream from Site A to Site B, beta diversity asks, how different is the community at Site B? How many species are new? How many are gone?
Okay, richness and turnover. But what about the interactions, the food web itself?
Right. For that, they used what's called a meta-web approach. They took the list of genera found at each site and combined it with existing ecological knowledge what these types of organisms are known to eat. This let them sort of computationally reconstruct the likely food web at each site.
And that lets you calculate things about the web structure.
Exactly. Things like how connected it is, how complex.
Okay, let's get into the results, starting with that richness data, the alpha diversity, because this is where you immediately see things aren't simple. The different groups, fish, bacteria, invertebrates, behaved quite differently.
Let's start with fish. Their diversity, their richness, largely followed the classic pattern. As you move downstream into larger river sections, fish richness significantly increased. More space, more stable conditions, generally more fish species.
That makes intuitive sense. But you mentioned a twist, a seasonal thing.
Yes, exactly. That increase in fish diversity downstream, it was significantly less steep in the summer compared to spring and autumn. The pattern flattened out.
Huh. Why would that be?
The paper suggests it could be things like higher water temperatures or lower dissolved oxygen downstream during peak summer, potentially creating less favorable conditions that limit diversity temporarily.
Okay, now contrast that with the bacteria. They did something completely different, right?
Completely different.
Yeah.
Bacteria showed the opposite spatial trend. Their richness actually went down significantly as the drainage area increased. They were most diverse way upstream in the smaller headwaters.
Wow.
And temporally, like the fish, their overall diversity across the whole catchment was lowest in the summer.
So fish prefer downstream, bacteria prefer upstream. Wildly different spatial patterns. What about the invertebrates? the bugs and worms and things?
Well, the vertebrates basically threw spatial patterns out the window. Drainage area the size of the river section had almost no significant effect on their richness.
Really? So what drove their diversity?
Season. It was all about the season. They saw this major drop in invertebrate richness from spring into summer and autumn.
Why? Where'd they go?
They flew away. It's tied directly to their life cycles. Think about mayflies, caddisflies, dragonflies. Many aquatic insects spend their juvenile stages in the water, but then emerge as flying adults in the warmer months to reproduce.
The emergence, of course.
Precisely. So when those adults leave the water, the measured aquatic invertebrate diversity just tanks. It's a totally natural ecological event, but imagine if you only sampled in summer, you'd get a completely skewed picture of the invertebrate community.
Yeah, you'd think the river was almost barren of insects when really they've just, well, moved phase. That really highlights the need for seasonal sampling.
It absolutely does. Now, if we look at beta diversity, that community turnover metric, we see interesting patterns related to how these groups move around too.
Okay.
For fish and invertebrates, it was generally as you'd expect. The further apart two sites were along the river, the more different their communities became. Jacquard's dissimilarity increased with distance. Makes sense, right? Things change as you go downstream.
Yeah, that seems logical.
But the bacteria, again, were the exception. For them, this pattern of increasing dissimilarity with distance only really held true in the summer.
Only in summer. Why?
The thinking is that during spring and autumn, when river flows are generally higher, the water flows strong enough to effectively mix bacterial communities throughout the catchment. It homogenizes them to some extent.
So they get washed around more.
Pretty much. But in summer, when flows are lower, local environmental conditions become more important, and distinct bacterial communities can establish themselves in different places, leading to that stronger distance decay pattern.
That's a really neat example of how the river's physical force affects dispersal differently for microbes versus larger animals. Okay, so richness patterns are complex, turnover patterns are complex. Now let's layer the food web on top. This is where you said it gets really interesting. They looked at 4 food web metrics: link density, connectance, nestedness, and omnivory. Maybe quickly define those for us. What do they tell you?
Sure. Link density is straightforward. It's the average number of feeding links per species or taxon in the web. Connectance is a bit more abstract. It's the proportion of all possible feeding links that actually exist. A highly connected web is often considered more robust.
Okay. And nestedness.
Nestedness describes a particular kind of structure. Imagine Russian dolls. In a highly nested food web, the species found at sites with low diversity are basically just a subset of the species found at the most diverse sites. It implies an ordered structure, often related to efficient resource use.
Right, and omnivory.
That's eating at multiple trophic levels. So an animal that eats both plants and other animals would be an omnivore. Higher levels of omnivory can sometimes indicate a more complex and resilient food web, as predators have alternative food sources.
Got it. So what did these metrics show spatially moving down the river?
Spatially, they found that connectance, nestedness, and omnivory all tended to increase as you moved downstream towards the larger drainage areas. This was especially noticeable in summer.
Wait a second, let me make sure I heard that right. Food web, complexity, connectance, nestedness, omnivory increases downstream most strongly in summer.
Yes.
But didn't we just establish that invertebrate richness, a huge part of the food base, is lowest in summer because they've all emerged and flown off? How can the web be more complex and connected when there are fewer species around?
That's the core finding. That's the disconnect. Richness is just a count. Structure is about the interactions. Even though many insect types might be gone in summer, the organisms that remain, the remaining invertebrates, the fish, the bacteria, are often at their peak metabolic activity in the warmer water.
Okay.
They're feeding more, growing faster, interacting more intensely. So the potential number of active feeding links, the complexity of the interactions among those present, can actually increase even if the total number of species is lower.
That's counterintuitive, but makes sense when you explain it like that. It's about activity levels, not just presence-absence.
Precisely. But then look at the temporal patterns across the seasons. Season had a strong effect, particularly in autumn. Link density was highest in spring, but all 4 metrics, link density, connectance, nestedness, and omnivory, were significantly lowest in autumn.
So the whole food web structure kind of contracts or simplifies in autumn, even though species richness wasn't necessarily at its absolute lowest then?
Exactly. Summer had the lowest invertebrate count, but autumn showed the lowest structural complexity. This suggests a real shrinking of the functional food web as the system heads towards winter, maybe due to falling temperatures and declining primary production. The connections themselves seem to weaken.
Which again hammers home that point. Richness in one season doesn't predict functional structure in another.
Mm-hmm. And they looked at functional diversity too, the variety of feeding roles, like shredders, grazers, predators.
What did that show?
It showed a slight, marginally significant decrease as you went downstream. So functional diversity was actually highest upstream.
How does that fit in?
Well, it aligns pretty well with the classic ecological idea called the river continuum concept, the RCC. The RCC basically predicts that headwaters, the small upstream breaches, get a lot of their energy from external sources, leaves, twigs falling in from the forest.
That's the alloxinous material you mentioned.
Exactly, allochthonous meaning from outside. So you get a
Yeah.
diverse community of specialists there, like shredders that break down leaves. As the river gets wider downstream, it becomes more reliant on internal production algae growing in the river, which is autochthonous material.
Meaning from within.
Right. And that tends to support fewer or perhaps less diverse functional feeding groups overall. So seeing higher functional diversity upstream fits that model.
Interesting. And one more metric. Functional redundancy.
Yeah, that's basically asking how many different species or genera can perform the same ecological job. They found functional redundancy was significantly lower in summer compared to autumn.
Okay, there's a lot there. But the main message seems crystal clear.
It really is. What this study powerfully demonstrates is that you have these inconsistent patterns. Group-specific biodiversity, overall food web structure, functional characteristics, they don't all march in lockstep across space or time. You absolutely need to monitor multiple facets together.
You can't just pick one indicator like fish diversity and assume you know the whole story.
Definitely not. If you want effective, targeted conservation, you need this kind of joint, multifaceted approach.
Now, we should be fair and touch on the limitations. No study is perfect. They mention a few, right? Like defining functional groups at the genus level.
Yes, that's one. Assigning a single feeding type to a whole genus is a simplification. An individual insect larva might change its diet as it grows, for instance.
And they didn't include everything. Algae were left out.
Right. Algae are key primary producers, the base of the autochthonous food web. And terrestrial inputs like insects falling in or subsidies from the land weren't explicitly included either, though they're important.
And there's always the eDNA question of false negatives. Maybe a species was there, but at levels too low to detect, especially in colder water when metabolism slows down.
True. That's an ongoing discussion with eDNA. But the authors make a reasonable point that very low abundance or inactivity in cold seasons means those species are, functionally speaking, relatively absent from the active ecosystem at that time.
Fair enough. But even with those caveats, the significance seems huge.
Oh, absolutely. This is really a landmark demonstration. It shows eDNA isn't just for making species lists for a river. You can use it to get at these complex network-level patterns like food web structure across large scales.
It really opens the door for a new kind of monitoring, doesn't it?
It really does. This kind of standardized, high-resolution sampling could allow for long-term, repeatable monitoring of how entire river communities and their interactions are changing, not just in Switzerland, but potentially worldwide. It shifts the focus from, is species X present, to how is this whole ecosystem actually functioning?
So wrapping up, the big takeaway for you listening is that river ecosystems are incredibly dynamic mosaics. Different groups like fish, invertebrates, and bacteria respond differently to the river's gradient and the changing seasons. And crucially, the number of species doesn't directly predict the complexity or stability of their interactions.
Which leads us back to that need for joint study, and maybe a final thought to leave you with. Consider this, the study found food web structure was weakest in autumn, but species richness, particularly for invertebrates, hit its low point in summer.
So if our goal is to conserve the functional integrity of the river, understanding that autumn decline in structure is vital. It makes you wonder, doesn't it? How many of our past conservation decisions, based on single-season surveys or just tracking one taxonomic group, might have been inadvertently misleading us about the true health and resilience of these vital ecosystems?
That's a really powerful question. It suggests we might need to fundamentally rethink how we monitor and manage our rivers based on this deeper, more integrated understanding.